Good morning, ladies and gentlemen. Dear Professor Karadenis, thank you very much for your kind introduction. First of all, I would like to express my gratitude to Professor Schaltert, who invited me for this webinar. And I was several times in your beautiful country. This time thoughts about beta cell regeneration, unfortunately online, but hopefully next time we will be able to see each other again face to face. Ladies and gentlemen, when we talk about beta cell regeneration, or when we talk about type 2 diabetes at all, we know that type 2 diabetes is a disease of progressive beta cell dysfunction. And usually we know that in the time of the diagnosis of type 2 diabetes, we know that only 50% of beta cell function is still working. On the other side, we know that beta cell failure or beta cell loss in type 2 diabetes occurs as a combined consequences of high glucose and free fatty acid in circulation. On the other side, due to adipocyte secrete factors, chronic activation of immune system, genetic components, and many other different mechanisms. There are several candidate genomic variants that contribute to the risk of type 2 diabetes, and a number of these variants are located in genes that are known to regulate beta cell function and development. On the other side, besides genetics, we know that epigenetics is a normal gene regulatory phenomenon involved in human development of cellular differentiation. And we know that each cell type is in the body is genetically identical, but needs to differentiate phenotypically to sustain normally functioning human body like neurons, skin, and other cells as well. Epigenetics mechanisms refer to functional changes to the genome that do not involve any alteration in nucleotide sequence. So epigenetics mechanisms will not change nucleotide sequence in genes. And it's very well known that epigenetics is very important. And there is evidence that several external factors can impact gene function epigenetically, no matter positively or negatively. Histone modification are important in terms of the epigenetics, and also the DNA methylation and histone modification can be active during fetal life, but also in postnatal and adult life period, and can impact the level of expression of selected genes associated with type 2 diabetes. And we know that environment can alter the pancreatic islet epigenome and can affect beta cell function. So, for example, impaired intrauterine environment can result and can increase the risk of postnatal metabolic diseases such as decreased beta cell proliferation, mass, or even insulin secretion. On the other side, beta cell loss in response to nutrient excess and stress were traditionally felt or meant to occur exclusively via beta cell death or apoptosis. And we know that apoptosis can occur due to high glucose level or due to glucotoxicity, due to lipotoxicity, but also due to many factors secreted by adipocytes, like pro-inflammatory cytokines, TNF-alpha, interleukin 6, leptin, and many other things. And also innate immunity and autoimmunity can play important role in beta cell apoptosis. But it seems that beta cell apoptosis or beta cell death is not the most important thing in the process of beta cell loss. And a few years ago, it was published a very nice and elegant review about beta cell differentiation in type 2 diabetes. In that period of time, I asked myself, dedifferentiation. What does it mean, dedifferentiation of beta cell in type 1 or in type 2 diabetes? And it seems that beta cell dedifferentiation could play important role exclusively in diabetes, but also not just in type 2 diabetes, possibly even in type 1 diabetes. And we know that beta cell dedifferentiation can be due to FOXO1 deficiency during pregnancy and aging, and it is primarily associated with beta cell dedifferentiation rather than death. And just would like to remind you that FOXO proteins are a subgroup of forehead family of transcription factors. So it means proteins that can bind to DNA. And you can see it here that FOXO transcription factor can be located in cytoplasm or in nucleus. If they are located in nucleus, they can target gene expression and can switch on genes. And FOXO1 is transcriptor factor that plays important role in regulation of gluconeogenesis and glycogenolysis by insulin signaling, and could also play important role in decision for pre-adipocytes to commit to adipogenesis. You can see it here. If FOXO1 is located in cytoplasm, in that case, it doesn't play important role in transcription or switching on or switching off some genes. But in terms of mild hypoglycemia, and it will be located in nuclear, which can be important for the signaling, insulin signaling, and also producing insulin. But in severe hyperglycememia, maybe even during aging or type 2 diabetes, will be located will not be located in nucleus, neither in cytoplasm, and that will be dedifferentiated beta cell, and we can call that non-beta cell, non-beta endocrine cell, which can have completely different role in pathogenesis of type 2 diabetes. So if we have FOXO1 here in cytoplasm, there is enough insulin. In nuclear, it will be losing possibility to secrete insulin, but in severe hypoglycemia, during aging or in type 2 diabetes, no FOXO1, and it will be dedifferentiated beta cell, which will at the end become non-beta endocrine cell. Sorry. So previously we thought that normal beta cell in some pathophysiological mechanisms, different pathological mechanisms, when they become dysfunctional, will be due to process of apoptosis destroyed and will disappear. It is not current theory. It was theory 10 or 15 years ago. But right now, the new hypothesis is that normal beta cell, when become challenged or they able to produce insulin, and dedifferentiated or reprogrammed beta cell will not produce insulin anymore, but we will produce glucagon or somatostatin or some other hormones which can act even opposite in terms of insulin. So from that perspective, from normal beta cell, which produce and release insulin, the dedifferentiated beta cell will not be able to produce insulin, but will be able to produce glucagon, somatostatin, and other hormones which act completely opposite or contra-regulatory to insulin. What is the trigger of beta cell failure? So there are many triggers for beta cell failure. That's the reason why we are not talking about beta cell loss or beta cell death, because right now we know that some beta cell can become only dysfunctional. Some beta cell can be dedifferentiated to non-beta cell. But so that's the reason why we usually use beta cell failure as a term. And the triggers of beta cell failure can be glucotoxicity, lipotoxicity, due to a high level of isolated amyloid polypeptide, due to different cytokines, which can cause the beta cell failure. But of course, as a clinicians, we are the most interested how to find the appropriate treatment for type 2 diabetes. And if we talk about beta cell dedifferentiation, in that case, it's of interest would be to find the therapy for redifferentiation to somehow to cause dedifferentiated beta cell to be redifferentiated again to functional beta cells. And there is a long history of investigation into pancreatic regeneration going back nearly a century. And we know that animal studies have suggested that exocrine pancreas possesses the intrinsic capacity for regeneration. But unfortunately, it's mostly in animal studies. By contrast, the endocrine islets have limited regenerative capacity in humans. And there is an increasing consensus that regenerative medicine approach would be helpful in type 1 diabetes, but possibly even in type 2 diabetes, in which there are substantial beta cell loss. And there are several reasons why we can believe that it would be possible. Islet hyperplasia is observed during pregnancy, during obesity. For example, during pregnancy, beta cell mass increases by 3 to 5-fold. On the other side, a high-fat diet-induced obesity can be accompanied by impressive increases in islet beta cell mass. But on the other side, also experimentally induced insulin resistance can increase beta cell mass up to 10-fold. So there is a long-standing hypothesis that even pancreatic stem or in human pancreas, and there are huge advantages in our understanding and enhancement of regenerative strategies. For example, from pancreatic duct cells, acinar cells, and centroacinar cells, which can be differentiated into islet endocrine cells, alpha cells, beta cells, or even other cells, delta or omega epsilon cells as well. When we talk about therapy for diabetes, oral and injectable anti-hyperglycemic therapy today, which we are using very common in everyday clinical practice, can have different extraglycemic effects. And you are all aware about extraglycemic effects of GLP-1 receptor agonist and SGLT2 inhibitors. We know that some of them can have beneficial effect on cardiovascular risk reduction. Also, they have beneficial effect in weight loss and also in blood pressure management as well. But when we talk about beta cell dedifferentiation or transdifferentiation, it seems that GLP-1 receptor agonists could have a beneficial effect in beta cell regeneration as well. Glucagon-like peptide 1 can increase pancreatic beta cells, and alpha cells could be a potential source for a new beta cell generation. So it seems that GLP-1 receptor agonist could be important in beta cell regeneration throughout maybe even alpha cell transdifferentiation. The GLP-1 receptor agonist action is more than just glycemic control, and you are all aware of it. But when we talk about effect on beta cell, it seems that GLP-1 receptor agonists could have beneficial effect on beta cell proliferation, beta cell neogenesis, but apoptosis. So from one side can suppress beta cell apoptosis and could increase beta cell proliferation. Several mechanisms are proposed, but it would be too into details. It wouldn't have sense for this lecture in this moment, because there are so many things which is also important to mention. SGL2 inhibitors also, it seems that they could have beneficial effect on beta cell mass and beta cell function. But it seems that it's a little bit different mechanism. I mentioned the potential mechanism of GLP-1 receptor agonist, but for SGL2 inhibitors, it seems that SGL2 inhibitors could improve insulin secretion in type 2 diabetes without decreasing of glucotoxicity. And we know that glucotoxicity is an important trigger for beta cell dedifferentiation, and it was found in different mechanistic studies with dapagliflozin, empagliflozin, in clamp studies, but also in OGT studies. But it seems that SGL2 inhibitors could decrease glucotoxicity, and amelioration of glucotoxicity is one of the possible mechanisms what could explain the beneficial effect of SGL2 inhibitors on beta cells. But of course, they can glucotoxicity, when we are decreasing glucotoxicity, also endoplasmatic reticulum stress and oxidative stress is also decreasing, what can have also beneficial impact on beta cell function and decreasing of beta cell loss. And also lipotoxicity might be also ameliorated by SGL2 inhibitors via improvement of systemic lipid metabolism. So it seems that decreasing of glucotoxicity, increasing of lipotoxicity, and also beneficial effect on decreasing of oxidative stress and ER stress as well could have the reasonable explanation for beneficial effect of the SGLT inhibitors on beta cell function and beta cell loss. I mentioned several times glucotoxicity. Glucotoxicity is a term which we can find in literature for several decades, and it means that high blood glucose level will cause many different effects on different organs, and glucotoxicity in long-term period will cause chronic complications. But in acute glucotoxicity can cause acute complications of diabetes as well. But it seems effect on beta cell function as well, and also on beta cell. So it seems that glucotoxicity can reduce the insulin gene transcription and also insulin secretion by itself. On the other side, lipotoxicity or free fatty acids level high could have also very detrimental effect on beta cell function. And we know that that free fatty acids are important because free fatty acids could have a beneficial effect on beta cell function as well, because they are crucial to maintenance of glucose-stimulated insulin secretion. So free fatty acids are important for glucose-stimulated insulin secretion, but chronic exposure of high level of free fatty acids could have opposite effect and could have detrimental effect and could impair insulin gene transcription, and also could impair glucose-stimulated insulin secretion and could eventually cause beta cell loss as well. But looking to the future is something what is even more important. We know that in the time when we diagnose diabetes, it's usually we thought that 50% of beta cell mass is already lost due to process of apoptosis. We know that beta cell type 2 diabetes is not just insulin resistance, and also it's not just beta cell death. We know that the mechanism of type 2 diabetes and pathophysiological mechanisms of type 2 diabetes are much more complex. But we know that beta cell function is still very important, especially when we choose the therapy for type 2 diabetes. And it's important to preserve beta cell if we can. And we know that in the past we had only few therapeutic options including sulfonylureas, for example, which will have detrimental effect on beta cell loss even after diagnosis. So for us, it's important how to maintain beta cell function as long as we can. And from that perspective, maybe we can modify disease. And there is no doubt that weight reduction can have beneficial effect, because weight reduction will improve insulin secretion and will cause and will decrease insulin resistance. So if we will, with weight reduction, we will increase insulin sensitivity, and with glucoregulation, we will avoid glucotoxicity, lipotoxicity, and all other mechanisms which can have a detrimental effect on beta cell loss. But also, of course, we have to emphasize the importance of modern therapy as well. And when we choose therapy for type 2 diabetes, it's also very important which therapy we will choose, because with some therapy we can preserve beta cell for a longer period of time, and also potentially GLP-1 receptor agonist and SGLT2 inhibitors could have beneficial effect on beta cell redifferentiation. It seems that GLP-1 receptor agonists are the best candidate for beta cell regeneration in the future. But also SGLT2 inhibitors and even insulin could have beneficial effect on beta cell redifferentiation, or at least suppression or decrease of beta cell loss due to decreased lipo and glucotoxicity. Of course, we have to consider other regenerative strategies, and also stem cells of progenitor-based islet regeneration could play an important role, especially in type 1 diabetes, but potentially even in type 2 diabetes, even stem cell-based immunomodulation, islet transplantation, artificial islet. So we have to think about all these things, but it's very important when we have a patient with type 2 diabetes, we have to consider which therapy we will use, because we can at least modify the disease, what will happen with type 2 diabetes in the future, and what will happen with beta cell. So there are so many things which I would like to share with you, because the mechanisms of beta cell differentiation or beta cell loss is much more complex than I had opportunity to mention in my lecture. Because pathophysiological mechanisms of type 2 diabetes are pretty extensive, and we still don't know many about beta cell loss in type 2 diabetes, and we don't know much about, and we don't have the whole picture about pathophysiology of type 2 diabetes. But I have to consider the time of my lecture, so unfortunately I have to stop right now, otherwise I will other speakers will not be able to deliver lecture in time. But ladies and gentlemen, I believe that I just briefly mentioned about the beta cell loss, beta cell dedifferentiation, and its role in pathophysiology of type 2 diabetes. And I am sure that beta cell dedifferentiation, or even more important, beta cell regeneration or beta cell redifferentiation will become the important issue in type 1 and also in type 2 diabetes in the future, to preserve the health of our patient, because our final goal is to have a healthy patient who will live, and to prolong their life with no complications. Thank you very much.